Assalamualaikum Prof. Ibda, selamat datang.
Waalaikumsalam warahmatullahi wabarakatuh.
Oke, mohon maaf lahir dan batin, Prof. Ibda masih bulan Syawal.
Maaf lahir dan batin.
Selamat siang, Prof. Ibda.
Iya, kemarin selamat siang. Ini sambil rapat PAK, jadinya dua ini.
Oh, dua ya.
Tapi udah selesai kok. Tinggal ini ngobrol sebentar.
Oke. Tadi sudah bisa keluar ya Mbak Itan?
Baik, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
The Honorable Professor Dr. Rifda Novalin MSI, The Honorable Dr. Isdi Sulistyo DIE, The Honorable Dr. Maria Diah Nurmainita MSc, The Honorable Professor Dr. Phil F. Schäberle, and Professor Dr. Insinyur Linawati Hardjito MSc as here, and all the audience. Good afternoon, ladies and gentlemen. My name is Riviani. It's a precious chance for me to be your master of ceremony on this very special occasion in international webinar about exploration marine bioactive compound. First of all, let's say thanks to Allah Subhanahu wa Ta'ala who give us guidance, happiness, healthy, and mercy so we can attend and participate in this special event without any obstacles. On this special afternoon, we have several agenda as follows. First, opening. Second, National Anthem Indonesia Raya. Third, report by Coordinator of Center for Maritime Biosains Studies, PSBM, Dr. Maria Diah Nurmainita MSc. And the next opening remark by Head of Institute of Research and Community Service, LPPM, Jenderal Sudirman University, Professor Dr. Rifda Novalin MSI, and a welcoming remark by the Dean of Fisheries and Marine Science Faculty, Jenderal Sudirman University, Dr. Isdi Sulistyo DIE, and the main agenda is presentation from 6 speakers. First speaker is Professor Schäberle from Justus Liebig University Giessen, and I am a Giessen, Germany. And the second speaker is Professor Dr. Insinyur Linawati Hardjito MSc from IPB University, and she is a founder of Asian Press. And the next discussion and awards certificate for all speaker and photo session, and the last is closing. Well, ladies and gentlemen, let's begin this event by reciting Basmalah. Bismillahirrahmanirrahim. To the next agenda, the song National Anthem Indonesia Raya. Please for the commit. National Anthem Indonesia Raya. Okay, move on to the next agenda, report by Coordinator of Center for Maritime Bioscience Studies, PSBM. The speech will be delivered by Dr. Maria Diah Nurmeinita, MSc. For Dr. Maria, time is yours.
Okay, thank you very much for the time. Good afternoon, everyone. Honorable Head of Research and Community Service of Jenderal Sudirman University, Professor Dr. Rifda Naufalin MS, Honorable Dean of the Fisheries and Marine Science Faculty, Jenderal Sudirman University, Dr. Isdi Sulistyo, DEA, Honorable Speaker, Professor Til F. Schäberle from Justus Liebig University and Fraunhofer Institute for Molecular Biology and Applied Ecology, Giessen, Germany. Honorable Speaker, Professor Linawati Hardjito from IPB University, and also Ocean Press founder, distinguished guests, lecturer, researcher, student, and all the participants. It is my honor to introduce myself. My name is Maria. I'm the coordinator of Center for Maritime Bioscience Studies, Jenderal Sudirman University. And this research center is one of the 17 research center in Jenderal Sudirman University, belong to the Institute of Research and Community Service. And our research center established in 2015, and we focus on maritime bioscience studies, especially in the southern part of Java Island, because our university is located in the southern part of Java Island. So on behalf of the Center for Maritime Bioscience Studies, I would like to express a warm welcome to all of you to the international webinar entitled Exploration of Marine Bioactive Compounds. We have to extend sincere thanks to the head of Research and Community Service Institute and also the Dean of Fisheries and Marine Science, because this webinar is a collaboration between the Center for Maritime Bioscience Studies and also Fisheries and Marine Science faculty. And I would like also extend my sincere thanks to the keynote speaker and all participants. Today, it is our honor for having two special keynote speakers. The first keynote speaker is Professor Til F. Schäberle from Justus Liebig University, and also Professor Linawati Hardjito from IPB University and Fresh Ocean founder. Today, Professor Schäberle will share about what he and his research group have been working on. They have been working on the marine natural products, especially on the heterologous expression and genetic manipulation of natural product biosynthetic gene cluster. And the second keynote speaker is Professor Linawati Hardjito. As we know, that Professor Linawati is a marine biotechnologist who worked as a researcher and academic staff at Department of Aquatic Product Technology, Faculty of Fisheries and Marine Science, IPB University. And also she's founder of Ocean Press. And this afternoon, she will share with us about her experience, how to develop the marine bioactive compound from the laboratory scale to the commercial scale. And I would like to report that totally there are 321 participants who are registered in this international webinar. And besides Indonesia, some of them come from Germany, South Korea, and also Japan. So this tremendous enthusiasm is a proof that exploration of bioactive compound is a very interesting and challenging topic. As we know that Indonesia is one of the countries which has the richest biodiversity, and it is often known as the mega diversity country. And also Indonesia is the largest archipelago country which has become a target of bioactive marine natural product chemistry research and number of the novel compound. So last but not least, I would also like to give my sincere thanks to the committee who have been working hard for preparing this event. And we do apologize for any inconvenience you might experience during this webinar. And once again, I would like to welcome you to our webinar. Please enjoy the fruitful sharing and discussion about the exploration of marine bioactive compound. Thank you very much.
Thank you, Dr. Maria, for the report. And the next agenda is opening remark by Head of Institute of Research and Community Service, Jenderal Sudirman University, Professor Dr. Rifda Novalin. For Professor Rifda, time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The Honorable Dean of the Faculty of Fisheries and Marine Sciences, Jenderal Sudirman University, Dr. Isti Sulistyo, His Excellency Professor Dr. Til F. Schäberle, Justus Liebig University, Giessen and Giessen, Germany, the Honorable Professor Dr. Insinyur Linawati Hardjito, LPPM University and Ocean Prize founder, the Honorable Dr. Maria Diah Nurmenita, PSBM Coordinator, Jenderal Sudirman University. The first, I would like to thank of our God that has given me a mercy and blessing that can stand here to give a welcoming speak here. Ladies and gentlemen, of this time, I present a welcoming speech that conducted in LPPM Jenderal Sudirman University in an international webinar by the title Exploration Marine Bioactive Compound. Here, the term of reference that will be included on the webinar. As we know, Indonesia as a country with mega biodiversity whose world value doesn't only stop at the variety of organisms that exist, but also the compound contained in this organism. And 2/3 of Indonesia's territory is the sea. Marine organisms in Indonesia are very diverse and have high potential that can be utilized in the field of human health, agriculture, aquaculture, as antibacterial, antioxidant, antifungal, antiviral, and others. Bioactive compounds from the sea can also be found in microorganisms. The webinar was held to discuss bioactive compounds in the oceans, how they are used, and how bioactive compound obtained on a laboratory scale can be developed to commercial scale so that they can provide benefit to the wider community. Thank you for keynote speakers. Thank you for participants, students, lecturers, and researchers, and as well as the general public. thank you for your attention. I hope that this international webinar will add to our insights. Hope that the future we can advance Indonesia with knowledge and research that can provide benefits. We hope that you will enjoy a virtual webinar PSBM organized by the Research and Society Institute Jenderal Sudirman University. Maju terus, pantang mundur. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Rida, for the speech. And the next agenda is a welcoming remark by the Dean of Fisheries and Marine Science Faculty, Jenderal Sudirman University, Dr. Isdi Sulistyo DIE. For Dr. Isdi, time is yours.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. I hope my voice can reach all participants. Yeah, Bismillahirrahmanirrahim. In the name of Allah, most gracious and most merciful. Alhamdulillahirabbil alamin. Praise be to Allah, the cherishers and sustainer of the world. Let me ask you all to thanks to Allah for His blessings and His gift that we received until today. Our distinguished lecturer, Professor Till Schäberle from Justus Liebig University, Giessen, Germany, and Professor Linawati Hardjito from IPB University, Bogor. I would like to express my highly gratitude for your sincere voluntary and availability to be presenters and lecturers in this occasion today. The Honorable Head of Research and Community Service Institute of UNSOED, Professor Novalin, and the Head of Biosains Maritim Study Center of UNSOED, Dr. Maria Mainita. Last but not least, all participants, our colleagues, students, and all audience in this webinar. On behalf of Faculty of Fisheries and Marine Sciences of UNSOED, I would like to express our appreciation to this event, organized by very well by committee and especially by Bioscience Maritim Study Center under direction of Dr. Maria Mainita. I think, I suppose also that we all agree that our ocean or sea provide enormously benefits for our daily life, including bioactive compounds largely offered to explore and to exploit as undefinite matters. Recent sciences and technology or knowledge are discovered to do this, and as we all hope that Professor Schäberle and Professor Hardjito will share today. Thanks to this webinar that we can learn the newest methodology or technology in studying marine bioactive compounds. Therefore, please enjoy the seminar and do not hesitate to openly discuss during this highly valuable meeting. And welcome to all to this meeting, and we hope that we can obtain many benefits of this event. Thank you.
Thank you, Dr. Isdi, for the support and the speech. Well, ladies and gentlemen, we arrived. The main agenda is a presentation from all speakers. Before that, I will to introduce the moderator in this webinar. Her name is Dr. Diahruri Sanjayasari, MSc, and she's a lecturer of Aquatic Resources Management Study Program, Fisheries and Marine Science Faculty, Jenderal Sudirman University. And her background education is first the bachelor program at Jenderal Sudirman University and master program at ITB University and a PhD program at University of Auckland, New Zealand. Most of her work were understanding the physiological aspect on both marine and freshwater biota, which influenced by their environment. So let's recall Dr. Diahruri Sanjayasari, MSc. How are you today, Dr. Diahruri?
Alhamdulillah, I'm good. Thank you very much, Bu Rifi, for introducing me today.
Okay, and the next time is yours for Dr. Diahruri.
Yes, thank you very much. Okay, good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Alhamdulillah. First of all, let us send our praise to Allah Subhanahu wa Taala for all the bless, all the mercy, and all the joy that had given to us, so that today we can get it together here to have this international webinar, which held by PSBM. And unfortunately, we still have to wait Professor Schäberle to come to the meeting. And based on our information, it's supposed to be on a few minutes. Yes, ladies and gentlemen, today we have the privilege that in this meeting we are going to have two expert speakers related to the exploration of marine bioactive compound, because as we know, Indonesia is a very huge country and covered 2/3 of waters, which is oceans, and there are so many secrets in the oceans that we need to explore, especially the bioactive compound. And the first expert speaker that we would like to have today is Professor Dr. Til F. Schäberle. He is from Justus Liebig University, Giessen, Institute of Molecular and Applied Ecology and Biology, and Institute Molecular Biology and Applied Ecology is in Germany. And today, he would like to deliver his talk related to heterologous expression and genetic manipulation of natural product biosynthetic gene cluster. So this is his CV. Can we scroll down? Yeah, he lived in Giessen, Germany, and originally from Germany. Education mostly in Germany, and his research interest is related in. This is from Professor Schäberle. Please unmute me. So please, the committee, can you unmute Professor Schäberle? The chat, on chat room.
Yeah, thank you.
Okay, good afternoon. I'm sorry, what time is it in Germany currently?
Good for me, it's good morning. It's 9 o'clock in the morning.
Oh, good morning.
Good afternoon
morning,
to
Professor
Indonesia.
Schäberle. Yes, thank you very much for you're providing your time in joining this international webinar today. I think we had enough for the CV. Mas Iqbal, we can close it down.
Yeah, thank you very much already for the introduction. And I'm really happy to be here. You also have to give me the right to share the screen, then I can put on my presentation. And yeah.
Yes.
It's great to see that.
Before we start, yes, I'm sorry, Professor Schäberle. So before we start the international webinar, so ladies and gentlemen, all the expert speakers will deliver their talks from the first expert speaker to the second expert speaker, Professor Lina. And after that, we will have a time for discussion for about 30 minutes. And each of the expert speaker have a time to deliver their talk for about 30 minutes. Would it be all right, Professor Schäberle and Professor Lina?
Yeah, I think so.
Yeah, thank you.
Yes. Okay. So before we start, could you please share Professor Lina curriculum vitae? Yeah, Professor Linawati Hardjito is from Bogor Agricultural University. So her research interest mostly is about marine biotechnology and bioprocess. Maybe we can engage later. And if you're interested on her CV, you can directly ask her later on. Yeah, I suppose it will be all for the introduction of our two expert speakers today. Thank you very much for sharing the CV. And I think no further ado for the introduction. This is the time for the expert speaker to deliver his talk. The first one is from Professor Schäberle. The time is yours.
Okay, thank you. I don't know which screen you are seeing at the moment. Is it the presentation or the presenter's mode?
I think it's not the full screen yet.
Like that?
Yes, this is.
Okay, perfect. Okay, then thank you for the introduction. I'm very happy to present a little bit of our research today. Yeah, the title is already announced. It's about heterologous expression. So how to work with biosynthetic gene clusters if we already know the genomic information. Shortly introduction about our work in the group. We are doing natural product research. And maybe our mission and vision is to discover and design new molecules which open ways to improve and extend human well-being. We really start from the beginning, usually with the isolation of new bacteria, also doing a very classical approach most of the time, so bioactivity guided isolation of active compounds. And if we identify compounds of interest, we usually also look into the biosynthesis, into the biosynthetic gene cluster, how is such a compound produced, and then to see if this would be maybe also possible, what I want to tell you today, to make a heterologous expression, to take the whole biosynthetic information, transfer it to another organism to produce higher amounts. We also look into under-investigated habitats, which I think the marine habitat still is. And microbial dark matter means like bacteria which are not culturable to date. And this is also linked to this approach that we can do it culture dependent, if we can work with the microbes, or if it's independent, which means we only can use the genetic information. Okay. Let's move on to the talk today. I want to start with a general introduction about natural products, of course, with a focus on antibiotics, because this is also the focus of our work during the last years. And then how to link genes and natural products, because this is the first thing we have to do if we want to work with the biosynthetic gene cluster. And as an example, I brought you today Taobactin, which is a very promising antibacterial active compound. And there we go through the identification of the biosynthetic gene cluster, the heterologous expression, and also how such a compound can be modified. About antibiotics, I think in this audience, you are aware that resistance of the bacteria has really reached alarming levels. There are a lot of bacterial pathogens out there where only few, if any, available treatment options are left. And this report from the World Health Organization was also renewed. And I think it's also something like a pandemic situation. It's got developing a little bit slower, but it's really something we have to keep our eyes on for the next years. Of course, there are different things why antibiotic resistance is spreading so far. It is that we need antibiotic stewardship. This means how many antibiotics and when are they taken. Maybe there's too extensive use in veterinary pest management, but also, as you've seen on the last slide, also ourselves, we can ask us, do I really need an antibiotic to get rid of this cold or something like that? So therewith, since we need new antibiotics, definitely, natural product research becomes more and more important again. And I would say everything is based on the biodiversity. So this is our starting point, and this is really important that we maintain and secure biodiversity, because this translates into the genetic diversity or is based on the genetic diversity, so they are linked. But what is important, they translate into chemical diversity. And these chemical molecules, this is what is of interest to us. We can extract them and make biological tests. Here, classic antibiotic activity test, we see this inhibition zone where the test bacterium, which you can see here, is not growing. This means there's an active component on this filter disk. And then following a bioactivity guided approach, we can try to purify the extract to really come down to one or some more active compounds, which are responsible for the indicated bioactivity. Here are just some examples of antibiotics we were working in our lab. And then when we know the compound, we also can move on nowadays, since next generation sequencing is getting more and more faster and also cheaper. Also, academic institutions can do genome sequencing, identify the biosynthetic gene clusters, like these genes are responsible. And I think this is a very important step to move on to the translational research, that at the end, maybe from our initial finding there's an antibiotic activity, we can end up to a medicinal drug on the market. Because in this last step, which is quite expensive, and this cannot be done by academia, there we need pharma industry as well. I think it's important that we have the biosynthetic information to get them back into the boat, because then we can modify the biosynthetic gene cluster, we can make new compounds, we can also produce a little bit more of the compounds. If we really take, as I said before, the whole information, put it into another strain, which we can easily work with. And this is also linked to sustainable production, because if we think about compounds which maybe are identified from sponges initially, where we have a slow growing microorganism, it's not very sustainable to harvest all the sponges to get the compound. But mostly the compound is produced by symbiotic microbes. And if it would be possible to isolate the bacteria and produce it in a fermenter, then we can produce much more and in a more sustainable way. So how to link the biosynthetic information to the product? Yeah, I would say, let's start with the DNA, where everything is encoded in. This is transcribed to RNA. RNA is translated into protein, which already might be the active component, or it's the enzyme which is responsible to build up the molecule, which then is giving the biological or pharmacological activity. Now I want to stress the point that nowadays, since so much genome information is available, we already know like which genes are responsible for which enzyme, and therewith for which catalytic activity. So we can predict, if we know how the biosynthetic gene cluster looks like, how the natural product should look like. And this is called genomic mining. And the beauty of the system is that it also works the other way around. If we know the structure of the natural product, we could also predict how the biosynthetic gene cluster should look like. And this I called here reverse genetics. And this mechanism that we can give educated guesses like how it should look like and to link the genes with the product is especially true for modular systems like the polyketide synthesis and non-ribosomal peptide synthesis. Because there we have in each module like one building block is incorporated. And of course, it's also true for ribosomally synthesized and post-translationally modified peptides, so-called RIPs, because there directly the order of the peptide is reflected in the gene sequence. I want to exemplify that on a polyketide biosynthesis, this modular system, and basically everything what you can see here is one module. We have a loading module, then there's module 1, module 3, module 2, 3, and 4. And each of these modules is incorporating one building block. The building block is usually acyl-CoA. So let's put these units there, and then we have just different colors to see how it's in the growing chain, how it's happening. So this is attached here, and in the first reaction, this is transferred from this enzyme domain to the other one. So by the action of a ketosynthase, these two molecules are connected, and we get this nascent chain, which we now move on to the next module. Again, the action of the ketosynthase attached it here, and we have 3 building blocks. And in this module, we have additional keto reductase, which converts this keto bond into a hydroxy group. And now let's move on to the next unit. With all 3, again, ketosynthase, keto reductase, putting in the hydroxy group. Now we have an additional enzyme domain, the dehydratase, which is responsible to cleaving off the OH group, ending up in a double bond. Let's move on to the last module. Again, ketosynthase is putting it here. We are working on the blue unit, keto reductase, incorporation of a hydroxy group, dehydratase, double bond, now NO reductase, ending up in a single bond. And then this assembled molecule chain is released by the catalytic action of a thioesterase. So this was the principle behind it. And I think it's pretty interesting that nature just used this very limited set of enzymatic functions, and just by putting them together in different orders can create a whole wealth of molecules. And this is the same for polyketide synthesis as well as non-ribosomal peptide synthesis. Here are just amino acids incorporated, but the basic principle is the same. And then on this molecule, there are just some decorations, I would say. We can put a methyl group there, put a sugar there, put a halogen there, and so on. So in principle, this is an assembly line. And if we see here, like the acyl carrier protein is holding the chassis, is holding the molecule, and then the other enzymes, the ketosynthase, are putting something there and doing some modifications. There we have a methyl transferase, which is screwing a methyl group there. And there was, if we know how the assembly line looks like, how the genes look like, we can predict how the molecule looks like. And this is called the Co-Linearity Rule. This was for the modular systems. And of course, if we go back basically to the normal way from DNA to protein, the ribosomally synthesized ones, if we have this compound of interest, this AA means amino acid 1, 2, 3, 4, 5, 6, 7. We can look in the genome information if we can find information for these amino acids. And usually we can find this 7 amino acids somewhere within a gene encoded. Mostly the genes are bigger because there's also leader peptide encoded and sometimes also a tail. So it's somehow hidden within this gene, but we can find it. So this is here in this propeptide located. Then usually in a biosynthetic gene cluster, we also have like genes for the transport of the molecule to transport it to the outside, and genes for the encoding for the modification. In this example, we have a modification since it's building 2 rings here. And also here, the cool thing is that it also works the other way around. We can take the genes, transfer them in the heterologous host. So this should be our bacterium, where we put the genes in, and then this bacterium is able to produce our compound of interest, which would be this molecule. Okay, so So far to the background, now coming to the example Taobactin. Here I want also talk about the bioresource selection for antibiotic research. What is a good environment to look for potential antibiotics? There are different rationales to that. One is that we look into a competitive environment, because there the probability of success should be higher. There are a lot of microbes, a lot of organisms which interact with each other. There are signaling molecules, so they have biological activity. Maybe one organism is inhibiting the other to keep the space. And another point we should consider, if you look for compounds with antibiotic function, maybe it's of interest to look for habitats which are associated with higher organisms, because the compounds should have the same requirements as we use it for humans. So this should be used for compounds. But here in the graphics, there's an example from an insect larvae. So we have this caterpillar. Within there are living nematodes, and within these nematodes, there are bacteria. And these bacteria are producing compounds with desired properties. Because if antimicrobial compounds are produced by these nematodes, the bacteria must be non-toxic to the nematode. So if they are not killing the eukaryotic higher organism, and they spread well through the insect larvae, that they are everywhere there. So this means they have a good pharmacokinetics. Yes, translated in our application, we want to have low toxicity, good pharmacokinetics, and usually activity against gram-negative pathogens, because they are the most problematic ones. And the gram-negative bacteria that are common to the gut, also to insect larvae, or to the microbiome of the entomopathogenic nematodes, are also related to the gram-negatives in humans. So a compound was identified. I will show you the structure later on. Here we see the activities indicated. There's the activity of Taobactin, and here the one for ampicillin as a control. So the pathogenic bacteria, you can see here the escape strains, which are of highest priority to develop new antibiotics. Gram-negatives, where we have nice activity in the low micromolar range. And the cool thing for this antibiotic is that it's very specific, because against gram-positives, here as an example, Staphylococcus aureus, no activity is observed. And also against symbiotic gut bacteria, there's also no activity. So it's nicely specific. And if we put it to bacteria, this is the E. coli cell, you see over the time that the cell somehow disintegrates. So in a video, it looked like that. The cells are proliferating, and then you see that they are lysing, they are plopping away. So it's really a bacteriolytic antibiotic. And Taobactin disrupts the outer membrane and causes the lysis of these E. coli cells. So this was very promising, and we moved on to a in vivo model, where we can see the minimum curative dose was investigated. You can see here in black. So it's a mouse model. The mouse were infected with this E. coli. If there was no treatment, in black, all mice died within 24 hours. If they are now treated with Taobactin, you can see nice dosage dependent. If there's 1 milligram per kilogram, there is already an effect, but mice are still dying. If 2.5 milligram Taobactin are given, all mice survived happily, exactly like in the positive control, gentamicin. So this was investigated for E. coli, as well as for Klebsiella and Pseudomonas, and really the multi-drug resistant strains in there. So we had in vitro and in vivo activity against gram-negative pathogens. Then there was the interesting question, what is the target of this new antibiotic? The targets are well known. Usually we have cell wall, we have protein biosynthesis, DNA synthesis, but all these well-known targets couldn't give a hit. And if we think about the lack of activity against gram-positive bacteria, this was already an indication to us, maybe the target is on the outer membrane. Is it on the outside, which would be really, really interesting. But since we couldn't find the target, or basically the collaborators in the US and Kim Lewis lab couldn't find it, they were breeding resistant strains. If you give sub-inhibitory concentrations of an antibiotic to a bacterium, they somehow develop resistance. And then you can slowly increase the concentration of the antibiotic. Yeah, so this resistance development takes place. Maybe we should say it's really, in this case, it was really, really slowly. So it's no concern for further development, but this is also the way how we can look for the mode of action. Starting here with 2 microgram per milliliter to really high, we had a really high increase in the resistance level. Then these resistance strains were sequenced. And what was of interest, that there were always mutations in one gene and also more than one mutation in one gene. And this gene is BAM-A, which is the central component of the BAM complex. It's shown here. The BAM complex is located in the outer membrane. You can see here BAM-A, encoded by the gene I showed you before. And here's the cytoplasm. So here's the inside of the cell, and the nascent proteins go through the periplasm into BAM-A, are folded there properly. This is, so to say, the BAM complex are chaperones, and then are released into the outer membrane, which is here the case. If now Taobactin is added, it somehow interferes with this BAM-A. And this mechanism is not working anymore. The outer membrane proteins are not formed. And therewith, at the end, the outer membrane gets unstable and the cell slice. So this is the molecule, Taobactin, as we said, really identified by a classical screening approach, looking for the bioactivity of extracts. It has really promising activity specific against gram-negatives. You can see here, it's like, these are peptide bonds. So it's just 7 amino acids, which then are somehow connected to form here a ring and here a second ring structure. And it hits a novel target with BAM-A. And now let's move on to the identification of the biosynthetic gene cluster and the heterologous expression of that. So here we have the molecule, and we really looked for this sequence tag, because these are the amino acids, tryptophan, asparagine, tryptophan, and so on, one by one. And looking into genome information, we found a gene which was encoding for this protein here. And within there, at the end region, are exactly these amino acids of interest. So the blue part is this propeptide. Then we have also 3 genes, DAR B, C, D, encoding for ABC transporter, and DAR E is the essential modification enzyme. What is maybe also of interest, there are no designated peptidases, because somehow this has to be cleaved off here to get the molecule. But the borders of this whole biosynthetic gene cluster could be well defined, because there are other strains and other genomes available where these flanking genes are present, but there's nothing in the middle. So we really could say, okay, this should be the biosynthetic gene cluster responsible for the formation of the antibiotic. So we took the well, in the first step, we inactivated it and really could see then it was not produced anymore. But then we take the whole gene cluster, these 5 genes, putting them into a heterologous host, which is E. coli or Vibrio in this case, putting them into this, and then the E. coli and the Vibrio were able to produce the antibiotic. So I think this was a pretty clear proof that these genes are responsible for the production, and heterologous expression was successful in really known bacterial workhorses. What we also found out during that way, if we look at these 5 genes, basically only 2 were essential. We could go down to a minimal biosynthetic gene cluster only consisting of the precursor and this modification enzyme, which forms these ring closures, and we can produce Taobactin. And heterologous expression was established in different hosts. We were faster and reached higher yields than the natural producers, starting from 1 to 3 milligram within 14 days. The E. coli is already producing about 30 milligrams within 2 days, but this can be further optimized. This was only in shaking flask, and I think if we go to a fermenter, this would be even more. Then we have protocols at lab scale for the purification, but there we definitely need to get better. So during the purification, we lose a lot of compound. So this should be optimized. But what is interesting, I would say, that we know the biosynthetic gene cluster and it's a WIP, and maybe derivatization should be possible. If you think about these 7 amino acids, in theory, they can be exchanged to all proteinogenic amino acids at each position, which will give a ridiculous high number of possibilities. We tested that, and so these are now unpublished data. Not at every position it can be exchanged, yeah. And then also not everything, only the green ones are active ones. So we can lower the number, not in every position we change it. If we just say we can change it 4 positions, we will have 200,000 possibilities. And this somehow would be a good jumpstart for medicinal chemistry, because we can modify the compound and then thinking about further modifications which could take place at the molecule. We really can make a lot of derivatives and screen them also for their bioactivity. Okay, and now let's see if we can come to make a turn to the marine habitat. And yes, this is also possible for Taobactin, because it was first identified in these insect-associated Photobactus strains, but it can be also found in the cluster in Yersinia strains. Maybe let's call them associated with humans, but also in Pseudoateromonas and Vibrio. And the last 2 are marine bacteria. Just looking at the biosynthetic gene clusters, this was the first one identified. Then we found in Vibrio there's an additional transporter gene there. And in some of the Pseudoateromonas strains, there was additionally like this gene here in green. And we wanted to see if these strains are also producing the compound. So the first step, this is the extracted ion chromatogram search for the mass of Taobactin. Yes, it could be identified. When I could see they can produce the compound. Furthermore, they also could produce a derivative. Go back one, look at the number, there's 483, 482. But since these are double charged ions, there's a difference of 2. So this is a natural derivative of Taobactin. And then further moving on, don't go into detail due to time. Here we did molecular networking, so also LCMS-based analysis, where you can look for the ion of interest. The ND here is the mass of Taobactin, and nothing seemed to be related to that. But when we used relaxed conditions, we could find also another molecule which is related to Taobactin. This one is not related to it, this is only single charge. But there was another mass of interest, and when we looked into detail, we saw it. The fragmentation pattern looked like Taobactin. And what is the difference? A mass difference of 80 Daltons indicates bromination. So, and here is the picture that the master student was really happy about. This is a simulation by the program, how a brominated isotopic pattern of Taobactin looks like. And this is what we found. So it really indicates the bromination of Taobactin. At the moment, we don't know at which position the bromide is attached. We assume it's somewhere at this ring. But it's also a natural derivative, which I think it's somehow optimized for the marine environment. And also there we tested this with heterologous expression. So we just took the minimal biosynthetic gene cluster, these 2 genes, star A and E, putting this putative halogenase in there, and putting it into our E. coli platform. And indeed, here you can see the original producer in blue, and in red, also the heterologous strain was able to produce brominated Taobactin. Also, iodized version was detectable. So then there's a iodide attached, bromination, iodized, detectable fluorination and chlorination was not possible with this halogenase. And this is already everything I want to tell you today, what can be reached to analyze in the biosynthetic gene cluster. And in a summary, I would say the classical natural product research is still very valuable for lead discovery. Classical bioactivity guided approach was performed here with the heterologous expression. We get access to natural products, also to other natural products if we only find the biosynthetic genes. In the case of Taobactin, I would say it's the most interesting lead structure because it has good activity or interesting activity pattern, and it hits a new target, which is very interesting. And the genetic manipulation open ways to create, so to say, non-natural natural products, because we can modify the amino acids and create natural products which we cannot identify in nature. And therewith, I want to acknowledge the people who were involved in the first Taobactin publication. And you see, this was really a joint effort. So these are all the others, not our group. And then, of course, I also have to thank my group. This is a picture from the good old times where we were allowed to stand beside each other. This is now really, really big group, I would say, because it's my university and my Fraunhofer group together. And to emphasize the people, here's your lecturer, Rianti. Thank you for organizing this talk here. And who else was involved? Niels was in the beginning involved in identifying the biosynthetic gene cluster together with Louis of Taobactin. So they first found it out. And then it's an Indonesian project somehow. And then Selina did the heterologous expression and improved the expression yield. And Deva was working on this derivatization, like which amino acids can be incorporated. And therewith, I thank you for your attention.
Right. Thank you very much, Professor Schäberle, for the very interesting talk today. It's very new information to us. Thank you. Maybe, ladies and gentlemen, later on you can prepare the question if you would like to give a question to Professor Schäberle. Now we would like to move on to our next second expert speakers, Professor Linawati. Yes, you can start share the PowerPoint.
Good afternoon, ladies and gentlemen. Thank you very much for giving me a chance to share what we are doing here in IPB, especially, but generally in Indonesia. So I would like to share my presentation. Can you see it? Can you see Ibu?
Yes, it's good now. We can see it.
Okay. Thank you. Okay. What Professor Schäberle talked is about very deeply knowledge about how the bioactive compound was synthesized and manipulated. But here I would like to focus on kind of using the bioactive compound to develop various of products within Indonesia. So myself, I'm working on the Aquatic Product Technology Department, Faculty of Fisheries and Marine Science, IPB University. And I also founder of Ocean Press. So Ocean Press is small, medium enterprises who develop various of active compound, especially marine, into different of product, especially cosmetica. And right now, the product is already marketed within Indonesia, and some we try to sell overseas. So if we talk about bioactive compound, which is substances or metabolite that show biological activities, such as antimicrobe that was explained in very detail by Professor Schäberle, and then there might be also antioxidative compound and cytotoxic and inhibitor topoisomerase. The compound that we choose here is based on our previous research. We do a research in 1994, and we start establishing a company in 2010. So we also learn about inhibitor topoisomerase as targeting anti-cancer agent, and also anticoagulant from seaweed and moisturizing agent, and also skin brightening agent having activity as inhibitor tyrosinase. So if you look at this one, it's explained by the head of LPPM Universitas Jenderal Sudirman that Indonesia is on a youth biodiversity, but here we only focus on marine resources that's already farmed by coastal community, because we do direct extraction, we don't do chemical synthesis. So we want to make sure that anything that we get from the marine resources can be sustained in the long future life. So we focus on seaweed, and also for fish byproduct, like for we also produce marine hydrolyzed collagen, and also chitosan from shrimp shell, and we also doing with coastal plants such as mangrove, pandanus, and also various seaweed as source of marine mineral. So how we develop marine bioactive compound here. So we have various of bioactivity, antioxidant, antimicrobe. We have several development here. The first is cosmeceutical or cosmetic. This is a cosmetic containing a natural active ingredient for health skin. And for pharmaceutic, actually we don't work on this aspect because we know that to develop pharmaceutic agent it takes a long time. And in Indonesia we have a kind of president decree that especially for seaweed we have a roadmap that has to be fulfilled within 5 years. And we also working with agriculture using marine bioactive from, especially again from seaweed. So we use it for biofertilizer and also biocide, because you know that IPP is one of the best university within Indonesia in terms of agriculture. And last but not least, we also working on a biological process. Actually, it's microbial based hydrolysis to improve the yield and activity of various bioactive compound. Again, we use marine resources, seaweed marine resources. If you look at this, the photo on the right hand side, this is we apply a seaweed fertilizer for watermelon, and we hydrolyze the brown seaweed, and we use it for coloring agent for batik. You know that Indonesia is very famous with batik all over Indonesia. From different province, they have their own batik painting. So if we talk about bioactive based industrial development, so I can say that we can divide it into 3 different categories. First is pharmaceutical, the one that explained by Play. And if you see here, the years to develop is, I think it's more than 15 years, and also high cost investment, though we can expect return. Even, yeah, it's true, it's more than like botanical medicine or personal care. But the expecting growth is least compared to the others. The others, like botanical medicine, we can do up to 20%, and also personal care. But for pharmaceutical is up to 6%. This data I took from Kate and Leif in 1999. But this is still, I think, up to now, I feel that still suitable, at least to estimate or to use it as one of a decision-making process, which kind of product we're going to develop in Indonesia. Why we choose like botanical medicine or personal care? Because if you see that Indonesia has a long historical a background for botanical medicine. In Indonesia, we call as a jamu, and also personal care. So the all kingdom, they have always a terrestrial ingredient that they use for cosmetic, for the princess, and also for the family of the king. So this both botanical medicine and personal care, at least according to my opinion, is very useful for Indonesia, especially if we want to get the result in a short time. So we can develop this product. The maximum is 5 years. So talking about innovation. So I would like to share actually what we are doing here in Indonesia, especially in IPP, how we take the bioactive ingredient from the marine into the commercial product. So our National Development Agency, Bappenas, they says that innovation is a kind of invention, is our research, but should be end up with commercialization. So this, if we talk about innovation, it's mean we have to deal with commercialization. So this one example, as I mentioned to you, that's I am one of the founder of Ocean Press, the small medium enterprises who all the technology and product are based on research from IPP University. If you want to know further about this company, you can check oceanpresscv.com or also IG Ocean Press CV and also Civet Ocean Press. So this part of a development that we established within IPP, especially our department and our mangrove. We use it for bioactive compound, avoiding the sun as a sun protector, and also the integrated production of carrageenan, and also the formulation of their product for food and non-food, and also the Pandanus tectorius. We use it for natural color, and also it's just the antioxidative compound, and also anti-cancer against cell line A280. And extract, hydrolysis, and process, also processing, and also how to use brown civet for brightening agent. So we check all the active compound and we apply into our cosmetic product. And last but not least is how we develop a bioprocess using marine fungi to hydrolyze brown civet to get more color and also the yield for batik coloring agent. So how we do this to transform the research result into the community. So if you, I think all of you are very familiar with what we call is ABGC plus, yeah? Academici, business, and government, and also community. So university can be anywhere, but this one I focus on what we are doing is IPP or other research center. And this university is working closely with the industry, CV Ocean Press, and to develop a community, we are supported by various international NGO like IMEC UNSOED, RARE, is based on United States, and also the various of ministry like Fisheries and Marine Science, Ministry of Industry, and also local government. And the community that's as our target is mostly is coastal people or civet farmer or or other community living in the coastal area. So how we do this one? To transform from the bioactive compound into the market. So first we have to own the technology and what kind of product. So we have to determine the scale and the specification of product, and don't forget right now how you treat the waste or the effluent is very important in order to sustain the ecological aspect of the environmental. And also in our university, we are very concerned about to use the byproduct. It's not discharging into environment, but transforming into a product that has economic value, like chitin, chitosan from shrimp, shell, and also marine hydrolyzed collagen from fish skin. So the stability of product is very important. Also, we have to investigate this one, because if we want to produce like cosmetic, at least our product has to be stable within 2 years. So after that, we have to make a business plan, and we have to determine who is the market and how we can get the raw material and where's the location of factory, and then how much investment that we need, and also profit and loss and cash flow of the finance analysis and internal rate of return and so on. So these parameters are important for investor if they would like to invest in a product based on marine bioactive compound. And of course, after that, we have to do manufacturing. We have to get all the permit, and then to determine the scale and also how we treat the byproduct and also the effluent or the waste. Because to get all the permit, you have to establish like what we call is a unit for to treat the effluent of industry, and also to get a permit from your neighbor that they are happy if you establish a factory there and so on. And from this one, you have a product to be tested in the market. This is not fully commercial, but it's limited volume. So we would like to have the preference of a customer, and whether our estimated stability of product is fulfilled in the real life. And also we have to check the pricing is very important. We have to check the competitor in the market. So from this one, maybe we need to improve our technology and also our product. And then finally, we can do scaling up for market extension. So that's the cycle when we would like to do the commercialization of marine bioactive compound. But fortunately, so if you want to go to cosmetic, like the one that I show you, to go to the cosmetic, all the ingredient should be listed in international nomenclature for cosmetic ingredient. And fortunately, all the list of the product are adopted by Indonesian Agency for Food and Drug Administration. So this is it's good for us. I mean, it's good for researcher in Indonesia, and the time is relatively short compared to pharmaceutical. And also we have a long track record of background on developing what we call as a jamu and also traditional cosmetic. So this example for cosmeceutical bioactive ingredient. So we use it, I mentioned to you, The Pandanus tectorius, if you check on international nomenclature for cosmetic ingredient, is already there. As this is a mangrove as a source of vitamin C, and this is a seaweed, you can call. If you apply for cosmetic ingredient, you don't have to name exactly the compound, but you just mention like algae extract. It means the various extract of algae can be brown, red, and this is also the algae. So these various resources, marine resources, they have like antioxidant, this pandanus, and also antimicrobe is a brown seaweed. So anti-aging is marine collagen and tyrosinase inhibitor, and others is from brown seaweed. So this microbial process to produce active ingredient from brown seaweed. So we use it the marine fungi. We isolated the fungi from seagrass, and then we do the cultivation at the laboratory, and then finally we try to use it to hydrolyze brown seaweed. And after that, after a week, and then we do extraction, and finally we can improve the yield from, I think, 16% into 35 to 40%. The yield is very important for industrial aspect because we have to count exactly the production cost to be competitive in the market. So how we do this one? We have tried powder of marine biota, and then we can do direct extraction using water or ethanol usually, because this is the most safe for various of product that we develop. And then we also can do the microbial hydrolysis. We usually isolate the fungi or the bacteria from seaweed or from seagrass or from other marine resources, and then we do this one filtration and drying, and we have a crude extract. So in product like jamu or nutraceutical or cosmetic, we don't have to get the pure compound. We just use extract, but we have to know exactly what the active compound is and how much the concentration within the crude extract. So this also what we have done. We isolate the tyrosinase inhibitor. You know, the Asian women, they like to have a bright skin. They feel that if they have a bright skin, and then they look so beautiful. So this product is very, I mean, very marketable in Indonesia, especially. So they want to remove the dark spot and so on and so on. So we isolate this from brown seaweed, but we have two different product. The first one is without hydrolysis, is direct extraction, where the yield is very low. I think it's only 16%. It's not enough for industrial application. And then finally, we improve the technology by hydrolyze using the marine fungi and marine bacteria, and then after that, we can so this is the analysis without the extract, and then the kojic acid as a positive control, and also brown seaweed extract. This the one that we use, antimicrobial focus, the one that's Professor Schäberle. And then if you want to go on this antimicrobe, you have to check the regulation of Indonesian Agency for Food and Drug Administration. This is, you can see what the target microbe is, Candida, and also Pseudomonas, and also Staphylococcus. But if you want to apply the cosmetic product to PPOM, it's not necessary you mention the compound. But maybe if this is from seaweed and then you just set is a seaweed extract or algae extract. It's mean the various extract of algae. As I mentioned to you, could be brown or red algae or green algae. And chitosan, we also use chitosan. This is a byproduct of fisheries industry. This we develop from shrimp shell. They have a good activity against several bacteria. But again, if you want to develop in cosmetic industry, you don't have to mention that it has like you use it as antimicrobial. So you can apply as like foaming agent or moisturizing agent, but in the same time, within foaming or moisturizing agent, they have additional activity as antimicrobial. These are our products. So we have a facial wash. This is from mangrove, and this is from green seaweed. Facial exfoliator, we use marine mineral from calcareous seaweed. And this is collagen face mask is from fish skin. And this one we use it. We use also marine collagen. And this one is series of brightening using brown seaweed. All this one on the bottom line. And what we have here that the bioactive compound that has already in the market as a final product or as an end product is antioxidant from pandanus and mangrove as sun protector, anti-acne from chitosan, anti-aging from marine hydrolysis, collagen, fish skin, or sea cucumber. But we don't use sea cucumber because in Indonesia we are not yet successful in sea cucumber farming. So as I mentioned to you, Ocean Fresh and also we in Department of Aquatic Product Technology, we focus on the marine resources that are able to be farmed because we want to sustain our industry. So marine collagen and also seaweed for anti-wrinkle and a lot others. So this our station in West Sumbawa. So we use the effluent of the seaweed industry and also the bioprocess, the fermentation of seaweed. We use it for fertilizer or growth promoter. If you see this one, the seaweed on the bottom right hand side. So this is comparison between a seaweed without pure stimulation from brown seaweed, and this is wet. So this is the age is about a week, so you can see differences. The growth is much better. I mentioned to you this is the industrial application using the marine fungi and marine bacteria that we isolated from seagrass and seaweed to hydrolyze the brown seaweed to improve the coloring agent and also the yield, because this is very important for us in order to reduce the production cost. So the brand that we have is Ocean Fresh, and Skoro Algae is concerned about the ingredient. Mosca is focused on a brightening series, Ribamel, and also the Beauty Bite is focusing on a nutraceutical that containing the active compound of marine hydrolyzed collagen and also chitosan and also seaweed extract. Thank you very much.
Right, thank you very much, Professor Linawati. It's very interesting. So from zero to mass production in Indonesia. Okay, now, dear all participants, it is a time for us for discussions. Please activate your raise hand menu if you would like to give a question. Okay. First, we have Undip Mada. Yeah, please. You can unmute yourself and deliver your question. Oh, you cannot unmute.
Okay.
Okay.
Thank you very much for Dr. Professor Schäberle and also Prof. Linawati Hardjito for your incredible speech for today. I'm really enlightened for new knowledges for me. My first question goes to Prof. Thiel. Well, I would like to ask, how do you maintain and optimize the Taobactin production in your microbes? And then during your experiments in heterologous expression for the Taobactin, did you also isolate any interesting compounds or kind of Taobactin analogs with similar activity or antimicrobial mechanism of action? Because in my laboratory currently, we are also working with exploration of new antimicrobial substances from marine bacteria and fungi. And then we have a little bit problems or obstacles to maintain the production, because in the first or until third production, our microbes still produce similar compounds, but then after that production suddenly gone. And then even though we already checked in the TLC, still the interesting spot was there, but since we don't have any HPLC or like LC mass, so we cannot identify or detect the compounds well. And then for Prof. Linawati Hardjito, thank you very much, Prof. Lina, for your lectures. I'm one of your former students in THP IPB, and I always got inspiration from your lectures. And then a little bit interesting, very interesting for me, and a question for you, since you were my examiner when I was in undergraduate. So why do you still not working with microbes to produce certain compounds for cosmetic? Because as I listened before, that you use the microbes only for increase the yield of the pigment production for batik, if I'm not mistaken. Because as I know, there are a lot of active compounds from microbes, such as kojic acid. It's produced by Aspergillus. And then my second question for Prof. Lina as well is, as you mentioned before, Professor, that every compounds that we use for cosmetic productions, we need to, everything must be listed or get permission from BPOM. And because as you mentioned before, you only use the crude extract, how could you get the permission from BPOM? Because I'm also pretty interested maybe in the future to develop my research more to product derived from marine microbe. Thank you.
Right, thank you very much, Mada.
Who first?
We have a second. Yes.
Maybe
Okay,
I
okay.
start directly, because there was many questions by Mada. Thank you. I think I shortly go into the idea that, or the problem you have, that you are losing the activity. This is a very known phenomenon. If you have the bacteria like coming from the nature, coming from the wild, they are much more productive. And if you have them in the lab, that they are losing activity. So what is important, of course, that you directly, when you isolate the bacterium, really make a lot of cryostocks, that you always can go back to that. Because here, passaging on plates, on liquid, they're getting weaker and weaker maybe in production. But you only can, at the beginning, go back to this by medium variation, testing different growth conditions, yeah, to find the conditions that they are motivated to produce. In our case, with this Photorhabdus at the end, this is a relatively easy strain, because this can be cultivated in standard media. But also from the others, I mean, the yield in Pseudoadomonas is super low. And also the others were just identified in Vibrio, we saw it, or in Yersinia, we saw it on the genomic information, but we could not isolate it. So it's not every time switched on. I think it's really at the beginning, you only can do trying to modify the medium, trying to modify conditions, both temperature, different shaking, and play around with that.
Right. Is that answering your question, Mada? Hamida, can you unmute?
Yeah, thank you. And then thank you very much for the first answer, Prof. Still. And then for the second question was, during your experiments in heterologous expression for Taobactin, did you also isolate any interesting compounds or the analogs of Taobactin with similar activity or antimicrobial mechanism of action?
I mean, for the heterologous expression approach, it's very specific. We directly knew which genes we take, and there we only made Taobactin A. But then we could also modify that to make others. But this was like targeted by us, yeah. That we have also identified others genes which might be for Taobactin B, C, D, so different ones. So we can do this, but it's a very targeted approach. But of course, it's possible.
No. Okay. Thank you very much, Professor.
Right. Thank you very much. Now the second question for Professor Lina.
Okay, thank you, Mada. How are you doing? Okay, your first question, why we don't use microbe? You know that the Ocean Trust is dealing with the coastal community that maybe they are not familiar how to culture microbe, and if you use microbe, and then you have to be very careful regarding contamination of other microorganisms. This is the reason, because we want to empower coastal community that's right now, maybe in the future, right now, I think it's not the time yet to use the microbe. And the second, in terms of how to get the notification of Badan POM, if you use the microbe, not all microbes are listed in the international nomenclature for cosmetic ingredient. Lactobacillus, yes. But the first time you have to check the list of microorganisms that use in the cosmetic product. And all the list is adopted directly by BPOM. So this is make it easy. So if you don't find this and then find the similar resources, like the seaweed, you don't have to mention like topoisomerase inhibitor, the brightening agent, that they won't accept it. But if you mention like the extract of algae, they have a cluster, you know, algae extract. They define as various of extract of green, of red and brown seaweed. So this covered by those clusters. So this is actually is good for us, you know, it's good for us. You don't have to prove it what's the compound, but you assure that the compound is contain the active ingredient that you targeted. I think this is a bit tricky with Badan POM.
Thank you, Professor. Very clear.
Okay, thanks.
Okay, thank you very much for the answer. For the speaker, I have one question from the chat room for Professor Schäberle. So this is from Anindita Sabdaningsih. She asks, which one better for finding new molecule for genome mining or classical method? As we know that purifying a single compound needs a big effort. Okay, Professor Schäberle.
Thanks, Anin, for the question. I mean, this is a very tough one. It's nearly philosophic, which is the better approach. I would say it's still the classical one is much easier to perform. If you think about heterologous expression, you need a lot of things before. You have to do the genetic work. And then if you just look into nature and look for DNA, you don't know in which organism or which organism can produce it. So the chance that there's failure and that you don't detect anything is very high. So therefore, it's always a long way to come to the pure molecule, but I think it's easier when you start the classical method. But of course, what needs to be thought about, or is that you not only activity is new because most of the activities are already known. You need a good dereplication, so relatively early in the process would be nice to have LC-MS-based analytics or so to see, okay, is it a new compound or a known compound?
Yes. Thank you for the answer. Is that answering your questions? And Adindita? Okay, we have another question here from Harwoko. Can you please unmute him? Mite?
Thank you for the nice talk from Professor Thiel and Professor Linawati. I have a question to Professor Thiel. Schäberle, maybe you are still remembering me. I'm Harwoko, Riante's friend from Professor Cox's group, Düsseldorf. I have two questions regarding to the bottlenecks in natural product isolation. The first, if we have a new compound, probably very low amount or inactive also. And the second problem is when we have non-compound, maybe also they are nons, the replication problem and also low amounts. In the problem of low amount, maybe you have a strategy or how to apply the natural product isolation to have good enough new compounds and active one and with enough amounts. For example, you have mentioned in your lecture one of strategy about reverse genomic. Could you please explain more detail about the reverse genomic? And the second one also you have mentioned about the heterologous expression. What are the big challenge to apply this heterologous expression in the bacteria or fungi, for example? Thank you very much.
Thank you for the question.
Okay. Yeah, hi, Habo. I think one point is really the yield, which you mentioned, that you don't get enough of a compound. Also there, it's really hard to find the conditions. I mean, classical thing is doing molecular biology or microbiology, playing with the production medium and so on. Of course, if the structure is known already, then you can try reverse genetics because you can predict or look into the genome if the strain is sequenced. If your producer strain is sequenced, you can look if you can detect the biosynthetic gene cluster. And if you can detect it, then heterologous expression might be a solution. But also there, of course, if you have a very new strain, which is not so known, it's hard to say which would be a good host, a suitable host for heterologous expression. So I think it depends always on your starting organism. If it is one which is well investigated, where there are examples for heterologous expression or also already some genome data available, it's easier. If you start with a really new species, you just isolate it, then it's getting very complicated.
Right.
Yeah, we can also you can also write me an email or so to and we can try to discuss that further, because I think it's a very long point.
Yeah. Yeah. Okay. I will go. So Professor Schäberle is open his time to do the later conversation with you. So that would be great. Okay, ladies and gentlemen, is there more questions here? Okay, Mr. Walter Balanza, you should activate your right hand menu, sir, but it's all right. The committee, please, can we unmute him? Walter Balanza.
You're still muted. Let's wait.
Yes, let's wait. Yes. Oh no. Okay.
Okay. Sorry, I was a bit late, but I think I just want to ask two questions. One for Prof. Schäberle. So the question is, okay, I think it's very typical that it's hard to bring the natural product, especially say from sponges, into the market. you know, is just 4 or a few of them now, 4 compounds or 5 that make it through to the market. But if, for example, if we find something or a molecule which is very interesting, can we bring it to the market with the heterologous technique that you have?
Walter, thanks. It's also a good question. I mean, depends on the molecule. As also shown today, it depends also on the application. Yeah, if you go to really pharmaceuticals, it's a long application procedure which takes a long time with all the testings, and therefore really the compound must be very promising that you do that. I think heterologous expression is a tool to produce the compound, but also there we have to see, is it more feasible to do it like by fermentation, or maybe you have to go to ask the chemists for chemical synthesis. And I think all the next steps, if it can be brought to the market, all this toxicity testing and things like that has to be done anyway. There the resource doesn't matter if it's from fermentation product or chemically synthesized.
Oh. Okay, thank you. And for the next questions, I will interested in knowing that we can bring here is not even pure compounds, but so many. So we have just like from mangrove. My question to Prof. Forgot.
Lina?
Yeah. What about the process? Do we have the test on, say, like if we do in the drug test and we have this, say, first clinical test and second, and what about if in cosmetic? Do we have many steps like that or shorter step, and how we confirm the result we've got? So is it we just have the extract and then we can claim it and then bring it to BPOM or something like that, or is there more detail that we have to do so that I'll say the extract that we have can be acceptable?
Yeah, okay, thank you for your question. So, like for mangrove, you have to check first in the international nomenclature for cosmetic ingredient. What is the ingredient that's already approved? And then you targeted that ingredient. So by that, you know, maybe it's a saponin or something like that, so alkaloid, something like that, and then you have to check it. You have to check how much the concentration within the crude extract. So this is actually is your internal strategy to be able to produce cosmetic product that has the real activity. For example, it's 1% or 2% or 3. And after that, for sure, the PPOM needs the allergic test and also self-life. So usually we develop this in the lab. So we use our student, the mother of our student, the auntie of our student as a target
panelist,
yeah.
as a panelist.
Yeah, as a panelist. And after that, and then when you apply into Badan POM, it's online. It's online, and then you choose which like mangrove, or you choose like saponin or flavonoid, and then that's already listed in the international nomenclature for cosmetic ingredient. You cannot mention like mangrove extract, because mangrove extract is not in the listed, but the ingredient within the mangrove extract is in it.
Okay, so in your experience, how long does it take? Is it to bring it 2, 3 months, 4 months?
To what?
I mean, to bring it from the research into...
No, it's 5 years, 5 years. 5 years, because you first you have to make sure your test result from laboratory, whether they are safe. I mean, they don't contain like the E. coli or Pseudomonas or Candida and so on. And then after that, you which compound that you are targeted, and then to choose that one, you have to check in the INCI which extract that you can use represent of your target compound, and then you apply. Usually, if your company is already listed in the Badan POM, and then takes about 3 to 6 months. But the first time we applied that one, including for all the regulation, and then the permit takes about 5 years. But right now, if I want to develop a new product, maybe it takes about 6 months. After we finish with the laboratory analysis.
Okay, thank you very much for the answer, Prof. Schäberle and Prof. Hardjito. Thank you.
Welcome.
Right. Thank you very much for the answer for both speakers. And I have one question from the chat room for Professor Schäberle. So, what is the most hardest compound to get extracted from your research, and how it become the hardest one? Thank you.
Okay. Well, probably the question should go to the PhD students who really have to do the work.
Maybe Burianti can help.
And then it's always the compound you are just working on. I think this is also then the point usually if the yield is very low. So if the concentration of the compound is very low, then it's hard. So for example, for Taobactin, we are now looking for this brominated version. And I think there it's super difficult to get it. So we see it by LCMS, it is there, but we need the fermentation is not easy. It's only working in standing cultures, and depends on the vessels of the flask you use. And then it really takes a long time to get enough material for that. And I think, of course, in doing research, there's a lot of time you see first an activity and you cannot follow up on it. That there are some compounds which we have an initial finding, and if you try to purify it, it's gone. Maybe there are also like 2 compounds which work synergistically, and if you try to isolate it, you lose the activity. So you cannot follow up everything.
Right. I hope that answer the question from William. Yeah, William. Oh, there is one question more from Ratna Saridevi. To Professor Schäberle, could you mention to me the example for tuberculosis medicine from one of most from the sea? I don't know whether it's really related to the talk, but yeah.
Yeah. In TB treatment, I don't think at the moment there's something in development which is coming from the sea or from the marine habitat. Of course, TB is still also a pathogen of concern, and we have also resistance development against the standard treatment options. So I know there are some antibiotics in the development pipeline which are in clinical phases, but of course also there we definitely need some more to find it. The complicated thing is for the initial screening, not everybody can do a TB screening, because you need a security level 3 lab, and also the assay needs like 1 month. It's not like, oh, E. coli you make overnight, it's growing or it's not growing, and TB takes a while. So it's not the easiest thing to follow up in the lab.
Yeah, TB is quite hard. Yeah, not necessarily David. Right. Is there any more question from the audience? Any more? Right. If there are no questions, I would like to give a question to Professor Schäberle. It's really interesting talk related to heterologous expression, where you can express the gene from other bacteria, especially the pathogen, E. coli, Vibrio, and etc. and how you choose or the type of bacteria that you have to, what is it, that you would like to express to that kind of pathogen bacteria. What is the characteristic to choose the bacteria?
Okay, so to choose the host for heterologous expression, yes. Of course, it's relationship, so phylogenetic relationship between the bacteria, where it's coming from, the compound. And then you have to, the easiest thing first is codon usage. You have to keep in mind, like the GC content is different. And, or maybe is it from a gram-negative bacterium? It's easier to do in gram-negative. If it's from gram-positive, maybe you can try to produce it in Streptomycetes. So I think it's really a relationship between the bacteria. But of course, the number of good hosts is very limited. Yeah.
Should we also consider that the host of the bacteria is easy to culture than the original one?
Yes, I mean, the benefit of a heterologous expression would be if the host is easier to handle, that it's faster and maybe genetic modification is possible. The problem is if you have a new bacterium, you have no idea how to modify it genetically. And therefore, we are going back to E. coli where we know what to do.
Right. Right. Thank you very much. Okay. Is there any more question for our awesome expert speakers today? No? Okay, let me check for the next slide. Maybe someone raising their hands. Or maybe in YouTube. Is there any question in YouTube? All right. Right. So if there's no more question, I suppose it will be all for today's talk. So we would like to say thank you very much for both speakers, Professor Schäberle and Professor Lina Watihartito, who delivered the talk today. And as an appreciation of their talk today, we would like to give a certificate of appreciation for the speakers. So the first one, we would like to deliver the certificate of appreciation for Professor Dr. Schäberle. Thank you very much. Everyone, please give applause to Professor Schäberle. You can put the reaction with the clap hands. Right. Yeah. Thank you, Professor Schäberle, for joining us today.
Thank you for having me.
Yes, we really appreciate it. And the second certificate of appreciation is for Professor Dr. Linawati Hardjito, MSc. Thank you so much, Professor Lina, for giving the talk today and joining us today. Please, everyone, give applause. Yes. Yes. Thank you very much. And I think because ladies and gentlemen, we come to the end of the international webinar today. And yes, thank you for joining us today in this event. And hopefully you enjoy the seminar today, and what we have today can add some more information related to the new method, how you can explore the secret from the sea, especially the bioactive compound. Thank you very much, and high appreciation. We would like to deliver to both expert speakers today, Professor Schäberle and Professor Linawati. So I suppose it will be all, and we would like also to say apologize. We would like to apologize if during the event there are some inconvenience that you may experience. Right. Thank you to all awesome audience today. And I think this is a photo session now before the closing. So please, the committee, be prepared for the photo session. And all the audience, please turn on your camera and give your best smile. Okay. 1, 2, 3 on my count. Right. Next slide. 1, 2, 3. Please turn on your camera and give your best smile. Thank you. Next slide. One, two, three. Next slide. Yeah. 1, 2, 3. Next slide. 1, 2, 3. And this is the last slide. Right. 1, 2, 3. Okay, committee, I hope you have all those beautiful faces who joined the seminar today. And thank you very much again for today's international webinar. And thank you, Professor Schäberle, thank you, Professor Lina, and thank you for all the audience. And I suppose it will be all. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.